afternoon, ladies and gentlemen. We're back once again with another amazing science tutorial video. I'm Coach Spivey, joined with my son,
Jordan Spivey.
And if you haven't already, go ahead and like and subscribe to our YouTube channel so you don't miss out on more of our amazing science tutorial videos. But without further ado, let's go and jump into our video today, and we're going to be looking at genetic mutations one-on-one. So let's do this. Boom. Before we start covering gene mutations, let's go ahead and go over the normal sequence of coding DNA to making proteins. And we're going to look at this DNA strand right here, which is TAC, ACC, and ACC. And we're going to go ahead and transcribe this DNA to mRNA using this chart right here. So that T is going to turn into an A, the A is going to turn into a U, and then that C is going to turn into a G. So right here we'll have U, G, G, and then we'll also have U, G, G right here as well. And now we're going to translate this mRNA into amino acids using our codon chart or our codon wheel. So we'll start off right here. We have AUG, which is methionine, so that gives us MET. And then we have UGG, so UGG, which is tryptophan, and that's going to be TRP. And then since this one is also UGG, this will be tryptophan as well. Abbreviation is TRP. So that is the normal sequence of coding DNA to making amino acids to making proteins. And now let's go ahead and take about 30 seconds so you can go ahead and code this next one. And you can go ahead and pause the video now. On the last slide, we went over the normal sequence of coding DNA into proteins. But what occurs when something goes wrong in that process? And so that's what brings us into mutations. And so what are mutations? Mutations are inherited changes in genetic information or our DNA. And mutations cause a change in DNA, which changes messenger RNA, which changes proteins, and ultimately changes you. And there are two types of mutations. We have gene mutations and chromosome mutations. But in this video, we're going to go ahead and cover gene mutations. And so a gene mutation is a change in the order of DNA that make a gene. And specifically, we'll be looking at point mutations, which are changes in a single gene, which include substitutions. And substitutions can be silent, nonsense, missense, conservative, or non-conservative. And then we have frame shift mutations, which can be insertions and deletions. And so we'll be looking at those 3 point mutations, which are substitutions, insertions, and deletions. Take a look at point mutation substitution. And substitution mutation is when one base is replaced by another base. It may not be dangerous unless it changes an amino acid needed for the structure and function of a protein. And this is what happens in sickle cell anemia. So let's go ahead and take a look at this normal gene right here. So here's our DNA right here, and it gets transcribed into our messenger RNA. And then that messenger RNA go ahead and codes to make these amino acids right here. But let's look at this substitution mutation. If you notice, this is an A right here, but in our normal gene strand, it was a T. So now, when we go to code this DNA into mRNA, this A is going to turn into a U. So let's go ahead and identify both of these. This A turns into a U. And then when we come down here to code for amino acids, instead of it being glutamine like it was in this normal gene, now it's arginine. And so this can have a negative impact on making a protein because this amino acid may be needed for the structure and function of that protein. Now let's move on to point mutations, which are changes in a single gene. And we have two types of point mutations. We have deletions and we have insertions. And both of these are also known as frame shift mutations because they cause a shift in the reading frame of the genetic message. So let's go ahead and start off with deletion. And as the name says, it's a base is removed or deleted. An example of a deletion disease would be cystic fibrosis. So let's go ahead and take a look at this. So if we look at this DNA strand right here, UAC codes for tyrosine, but if you notice, this C has been deleted. So when the C gets deleted, it causes all of these other bases to shift down, hence the word frame shift mutation. And if we go down, let's go and take a look and see what happens as a result of this deletion. Instead of it being UAC, it is now UAG, which codes for stop. And remember, like we said earlier, this premature stopping of making this protein causes glycine and lysine not to be formed. And so instead of having a full protein, now we have a protein that is not fully developed, and this is what leads to a genetic mutation. Our second frameshift mutation is insertion, and this is when a base has been inserted into the genetic code. An example of an insertion mutation would be Huntington's disease. So let's go ahead and take a look at this example. If you notice up here at the top, we have UAC, which codes for tyrosine, and then we have GGG, which codes for glycine. But if you notice at the bottom, this A has been inserted into the genetic code, and when it gets inserted, it shifts all of the other bases down, which throws off this amino acid production. So if you look, UAC, now it's UUA, and now it codes for leucine instead of tyrosine. And if you notice, it was GGG, but now it's CGG, and now it codes for arginine instead of glycine. And if you notice, this G is stuck here by itself, because remember, this base has been shifted down. So now, when we have these amino acids that have been produced instead of tyrosine and glycine, it can cause an overall harm in the structure and function of this protein. And this is what can also lead to another genetic disease. As a review, there are 3 different types of point mutations. If you notice at the very top, here's the normal sequence of DNA to protein synthesis. But let's look at our 3 types of mutations, and we'll start off with substitution. And in substitution, one DNA base has been substituted with another one. So this C has been substituted with T. And what this can cause is a change in the production of an amino acid, which can lead to a genetic disease. And we have 4 different types of substitution mutations. We have silent, nonsense, missense conservative, and missense non-conservative. And if we move over to insertion and deletion, remember, these are both our frame shift mutations because they cause that genetic frame to be moved or shifted down. So if you look at an insertion, that means that an additional base has been added to this genetic frame or to this genetic coding, and it shifts all of these other bases down. And what this can lead to is to the production of the wrong amino acids, which leads to a genetic disease. If we look at frameshift deletion, notice that this G has been deleted, which causes this whole DNA strand to be shifted down. And when this happens, this causes the wrong amino acids to be produced, which can lead to another genetic disease. Let's do an overall review of gene mutations by looking at the following concept map. So point mutations are a type of gene mutation, and we have 2 different types of point mutations. We have substitutions and we have frameshift mutations. And remember, there are 4 different types of substitution mutations. We have silent, nonsense, missense conservative, and missense non-conservative. And then if we look at frameshift mutations, we have 2 types. We have insertions and deletions. Let's do a little practice. And you're going to use your codon chart or codon wheel to get the amino acid sequence and the type of mutation it is. Make sure to refer back to the original DNA and amino acid sequence to identify the type of mutation. So whether it's a substitution, insertion, or deletion. So here's that original DNA sequence that you're going to refer back to with each problem. Now what I like to do is I like to separate my codons into 3s so I don't get confused when I'm doing my mRNA and amino acid sequence. So now we have TAC that's going to So here's the original DNA sequence. Ladies and gentlemen, go ahead and take about 3 minutes to complete the following mutated DNA sequences. And make sure to use your codon chart and codon wheel, and also determine what type of mutation it is. So go ahead and pause the video now. Now it's time for your check for understanding. And you're going to use your notes and knowledge of gene mutations to answer the following. Ladies and gentlemen, once again, I'm Coach Spivey, sign off my son Jordan Spivey. And if you haven't already, go ahead and like and subscribe to our YouTube channel. And also check us out at our website at www.fathersoninnovations.com. And like I always say, make sure you have a wonderful, awesome, positive day. Peace.